Okay. All right. So, as Macron said, I want to share with you some results on what we've been doing on the so-called bacterial growth laws. And basically, what you're going to hear is that when you look at bacteria from some perspective, the behavior becomes very simple. Okay, and despite the very complicated molecular interactions that's underlying it, right? And so there's first just the description, the statements of the simplicity that's summarized in terms of number of growth. I'm getting some feedback, but then the other part, and then the second part of the story is going to be, well, how did that come about? Okay. So just to make sure we all start on the same page. I think no matter where you come from, what your background is, ultimately, what we want to do to understand a biological system, a living system, is we want to understand, we want to be able to predict, right, if we make some reasonable perturbation to our system, could be an organism, a cell, or whatever, we want to be able to make prediction on how it responds. This could be environmental perturbation in terms of nutrient, in terms of drug. It could be genetic perturbations, something you do to turn off a gene or something, right? So I hope everybody agree that that's our ultimate goal, right, in terms of understanding biology. Now, how do we go about this? As a physicist, when I came to biology, I was told to think about a problem this way. When you provide you give your system a perturbation, then the system is going to elicit a bunch of signaling cascade, right? And then the output eventually is driven by some gene regulatory response. And then in between, there's some complicated molecular interaction around it. Okay. In fact, I think many of us from a physical science are recruiting into biology to tackle this problem, right? So this problem, I think I would say, is really an extension of what I call a fundamental tenet of classical molecular biology. Right, that is, by studying this research program, right, by studying what's happening down here, we'll eventually gain some understanding of up here. Right? So started with the Jacob, the regulation of the lac operon, understanding what the system is doing. Okay, and this we have gained a lot, right, starting from nothing through this program, looking at interaction, looking at what's happening, making some appreciation of what's happening at the top. So call this the bottom-up paradigm of doing biology. Okay. Now, the problem is this is really complicated, right? And then after a while, our attention gets more and more and more focused on this complicated part. Okay? And gradually, we start to forget about the original question we're asking, which is what's happening at the top. Right? I experienced this myself when I first got into E. coli, and many people talk to share a similar experience. Right? Your day-to-day work focuses on the interactions. All right. Now, this program, as I said, has been very successful, but when it comes down to making predictive understanding of a biological system, predictive behavior, there's a problem. Okay? The problem is that there are a lot of interactions, and each interaction comes with parameters, right? And you're talking about, so when you count up the number of parameters in a bacterial cells, easily you have 1 million cells, and every protein will have several binding constants and kcat and things like that. And then when you talk about combinatory interaction, you're talking about phosphorylation that affects the properties, maybe interesting mutation, and that affects the binding. And this, you're ending with macroscopic number of parameters, okay? And we have no way in principle of measuring these parameters in vivo, okay? And so what do we do? Okay, fine, we can write down all equations. You can write down mass equation, mass action, we can write down single molecule with stochasticity and all that, but ultimately, what do we do with a system with, even 10 to the 9 parameters? It's a lot of parameters, okay? I would say we can weight parameters by weight, how many parameters, okay? Right, but then on the other hand, And anybody who has looked at try to solve quantitative system, really, if to understand the system, you really need, all we can understand is few component system. Okay, there's no, they're difficult to attack a system with many parameters. So there's a fundamental problem of this bottom approach when it comes to quantitative understanding, right? Okay, so let me maybe give one example to illustrate in a context, a very simple example in the context of biology. Suppose you have your favorite cells. Mine's E. coli. It could be yeast, could be cancer cell, whatever, right? And you have a protein being expressed, GFP, let's say. Okay, and you can find out the concentration inside the cell. Now I ask you, suppose this protein is not the expression is not regulated. Okay, just constitutive expression. And so I compare two situations where the cell is growing slowly, barely growing maybe, and another situation where the cell is growing rapidly. Okay, so in E. coli we can get cells to grow at 20 minutes doubling, right, or easily 200-minute doubling. And in the two situations, fast and slow, right? In each case, I can look at the concentration of a GFP, right? So I want to just take a quick poll. How many think the fast-growing state will be greener in terms of a green intensity, concentration of GFP? How many think the fast-growing state will be greener? A few. How many think the slow-growing state will be greener? Okay, more than the first group, but the majority don't have a. So how many think there will not be a difference? It will be the same. Nobody think it will be the same. Okay, but most people have no opinion. All right. Okay, so now keep in mind. So this, for anybody studying gene regulation, this is the baseline, right? This is the simplest case. Okay. The fact that nobody has an opinion is interesting. Okay, but let's see. Just kind of a go through thinking, see how we think about it, right? So let's say inside the cell we have so this gene has a number of copies. It could be on a plasmid, it could be multiple copies on a chromosome, right? So G copies, each is spitting out mRNA at some rate alpha. mRNA is being turned into proteins with the rate alpha P. It's being degraded with the rate beta M. And in the simplest case for bacteria, these guys, the proteins are mostly stable, and they're just being diluted at a growth rate lambda. Okay, you can make it more complicated by having protein degradation. Okay, so if you multiply these factors together, so we're interested in the steady-state protein concentration, right? And you write down rate equation for synthesis and degradation of mRNA, synthesis, degradation, dilution of the protein. You can solve these equations, and the steady-state protein concentration is product of the synthesis rate and the degradation rate, the cell volume, and all that, right? And you see, well, a growth rate appears here, 1 over growth rate. Okay, so maybe this is the reason why there are more people that raise hand, right, for the second scenario, because it's diluting faster. If everything else being equal, it's diluting faster. So why should we take for granted that everything else is being equal? Right? When the cells are growing at a different rate, many things change. Okay, so in fact, this is generically not the answer. Okay, and so you can think about when a cell grow at different rates, what changes? Many things change. Chromosome copy number change, ribosome concentration change, cell volume change, everything changes. Some of these factors are upstairs. Some of these factors are downstairs. Okay? So really, it immediately tells you, you cannot answer this question just by thinking about it. Okay? You need to know details to see whether it's going up or going down. All right? So this is really depressing. Okay? Now, there may not be a single gene inside the cell that's not regulated. Okay? But as I said, the unregulated state is the reference state for anybody who's looking at gene expression, right? Because that's, you see what your favorite factor effect it has with respect to the unregulated state. Okay? So it's a situation that says your reference state, you don't understand it. Okay? And then you may say, well, why do I care about the cell growing in different conditions? Indeed, for good molecular biologists, if they want to study gene expression, they study in a condition where nothing else is changing. The growth conditions are changing. The only problem is that the cell doesn't care about that, right? When you change the external environment, many things change, including the growth. A lot of times you have to change from growth to non-growth, or fast growth to slow growth, right? So when a cell formulates its response, it's taking care of, it's got all of this into its consideration of formulating a response, okay? But then we don't know what it's doing. So that was, right, so, and in general, of course, we're not just interested in the unregulated state, we're interested in regulated state, right? So then you talk about regulating expression, everything we're talking about here, plus regulation, the feedback and all that, just gets hopelessly complicated, okay? So the question is, it seems like even to understand the simplest system, we need to know everything about the cell, okay? And that's really depressing. Right? So this was just an example of the kind of the complexity, which is there's so many parameters we don't normally think about that it's going to affect everything we're measuring. So fortunately, as a physicist, right, on the bright side, we'll say, well, actually, in physics, we have dealt with this problem that involve lots of parameters. Are there any physics undergraduates in the audience? A few, right? So if I ask you, what problems you have, so everybody has solved the problem, right? That involves a mode of parameters. How about just a box of gas? If I'm trying to study gas, property of gas in this room, okay? It's got 10 to the 23 particles, and I know they follow, what's the dynamics? They follow Newton's law, right? And to solve Newton's law, I have to input initial conditions, the position, the velocity, and then run Newton's law, and then they collide, and then eventually, okay, I describe the cell, right? So this is one way to study the system, and it needs to know lots of parameters. Of course, there's the alternative approach, thermodynamics, a single parameter, right? The gas constant R. Right? So this is examples of tremendous dimensional reduction, right? From Avogadro number of dimensions to a few dimensions. So how does this come about? Okay, so it's very tempting to see if we can go from here to there. Well, we can forget about that, right? So the famous person, Mr. Boltzmann, who tried to do this, eventually committed suicide. So don't try it. Okay. So even for a simple system of a gas, right, going from Newton's law to ideal gas, it doesn't work just like that. Okay. Instead, historically, the way it was developed was that these two are developed as independent discipline. Right? People back then didn't know gas were made of molecules. Okay, and so they developed. They made a characterization of what aspects affect the property of a gas. So there was thermodynamics, there was Newtonian mechanics, and then what Boltzmann did was develop a statistical mechanics, which was a shortcut to go from Newtonian mechanics to thermodynamics. Okay, a shortcut in the sense that if you tell me the rules of the games here, well, tell Boltzmann the rules of the game, right? He'll tell you a recipe so that you can immediately fast forward to the final state of thermodynamics without going through or the molecules running each other. Right? And the key to this shortcut, I apologize for the appearance of mathematics, but the expressions are very simple. So in principle, so let's say that there's this thing called rho, which describes everything about this gas. In principle, it depends on every particle, the position, momentum, okay, of every particle in this gas in this room. But what the statistical mechanics says is this rho, all it depends on is not directly on all of the property of the particle, but it only cares about energy of the system. Okay, so it actually depends on all this 10 to the 23 variables through a very simple through a function called energy, which itself could be non-trivial. Okay, so through this relation, ultimately this dimensional reduction comes from the fact that the property of the whole depends on the many variables through a single variable energy. Now, of course, what we want to do is to say something about the whole from looking at the molecules, molecular interaction in biology. Okay, so I couldn't do this. I have a very simple lab, and was already quite a bit to study microbiology for a theoretical physicist. But so we study E. coli, let's look at the growth, but then along the way we also had to deal with this situation because there's still millions of interactions going on inside the cell, and then we look at the physiology. It's much simpler. Okay. And so it's addresses the problem of how to conduct molecular biology. We have lots of information here, but even for E. coli, find that there's a lack of a quantitative characterization at this level. Okay, so what my lab did was we spent a lot of time characterizing this part. So it's almost like doing the thermodynamics for the gas. Okay, and then once we have information on the two sides, then we're going to try to link them together. Okay. And I don't know what the words for statistical mechanics will be, but we just call it quantitative biology, all right? But basically, at least for this aspect we're looking at for cell growth, there's a similar kind of a dimension reduction, okay? And so this will come at the end. But again, so the details are not important, but there's a central function that controls cell growth. In principle, it depends on hundreds and thousands of molecular players, okay? But it appears that this is done through its dependence on a single variable. And it's a similar type of sort of a dimensional collapse as we had in statistical mechanics that allow us to say simple things about physiology. Okay, so that was kind of, hopefully in half an hour, we'll get to this part. All right. So, so what do you gain from knowing that? Well, so this is the kind of things we're able to do. So, for example, we study steady state growth. So we characterize its property, and then we can use these properties to study what happens during growth transition. So here's a case where E. coli is growing in a combination of carbon sources, mostly on glucose. It's growing fast. Okay, black is the growth curve. And then at some point, glucose ran out, so there's a lag, and it continues growing. Right? And then the various components of the proteome in the cells respond. The green is ribosome. The red is, say, catabolic proteins. They respond in a different way. Okay? So with this underlying sort of simplicity, we were able to derive a single equation, okay, without any fitting parameters. We can describe these behaviors during the transition. No fitting parameters, right? Just from steady state, and can predict the behavior during the transition. And you can switch the initial and the final state. So start from a slow state and go to a fast state. The exact same equation. Okay, just flip the initial and the final condition, and then it gives good predictions on the observed behaviors again for these quantities. Okay. This is an example of what you can do with this. All right, so let me start with growth laws. Okay, so back starting in the late 1950s, so 60 years ago, so people were doing very simple experiments. This was the results from Ole Moller's lab in Copenhagen. They grow salmonella in different medium. And for each one, they very patiently wait until it gets to exponential growth. Okay, and so each of these points is one exponential growth. Okay, and during exponential growth, they measure RNA of the entire culture. They measure total protein in the entire culture. Okay, and what's plotted is the RNA-protein ratio. And you see there's a very striking linear relation with the growth rate. The medium are all different. These are kind of simple carbon sources, amino acids. This is the rich medium, wind up parts, whatever. Okay. Now, what does RNA-protein ratio mean? Okay, so for exponentially growing bacteria, most of the RNA is in the form of a stable RNA, ribosomal RNA. Okay. So RNA is a proxy for ribosome, And protein is the major component of the biomass, and it's roughly proportional to biomass, proportional to the cell volume. Okay, so anything per total protein is like per volume. Okay, so this is basically a proxy for ribosome concentration. So ribosome content, ribosome concentration, is a linearly increasing function of the growth rate. All right, what does this mean? Well, Moeller, at that time, they were not doing too many measurements, and every measurement they have, think about it, and they try to formulate a quantitative kind of rationalization for these results. And this is the rationalization. Suppose all of the ribosomes in the cells are engaged in translation. Okay, and they know that most of the bacterial proteins in exponential growth are stable. 95% we know are stable. Then the rate of protein accumulation is just reflecting the rate of how much ribosome is working. Okay, so for exponentially growing culture, the rate of mass accumulation is just the growth rate times the total cell mass, protein mass. And the rate of a total ribosome output is just the rate of elongation, this gamma thing here, multiplied by the number of ribosomes, here measured in terms of mass of ribosome. I'm just explaining, rewording what Moeller understood back then, okay? And so if you set these two equal to each other, you see that immediately you conclude that the fraction of the protein that's ribosomal proteins, so this, I call this phi r, phi is a fraction, okay? Phi, the fraction of the protein that's ribosome is expected to be a linear function of the growth rate, with the slope being 1 over the elongation rate, okay? So when we first saw this, we didn't believe it, so then we varied the elongation rate, and then we saw that indeed for mutants, ribosomal mutants with different elongation rate, it was still straight lines but with different slope. So verified these sort of things. All right. So basically, then this is the same sort of understanding is that what the meaning behind, the biological meaning behind this line is that most of the ribosomes are engaged in translation to get this kind of relation. All right. Now, this result sat there for about 50 years, and so about 10 years ago, we serendipitously came across this. I said, well, why haven't people done more with it? It seems like a great relation that's independent of the details of what food source the cells are growing up. Then thinking about it, we realized that it's actually missing a companion relation. Okay, so what I mean by this is this. So in this story, there are two things that determines how fast cell grows. One is the nutrient that you feed the cell. So this is what the parameter being varied. You feed it with different nutrient, the cell grows different rate. Another is how fast ribosome elongate. So in this experiment, they vary the nutrient, and the ribosome is just basically the wild type, whatever you have it, okay? And you get relation. But you don't know how it depends on ribosome the property, okay? So then we did a complementary experiment, we'll call the orthogonal perturbation. That is, for a fixed nutrient, we change the rate of how fast ribosome works. And that can be done by putting an antibiotic in the sublethal dose. Okay? Well, we got another straight line. Okay, so this is starting with glucose. This is no antibiotic. You start to feed it adding to the medium different amounts of sublethal dose of antibiotic that inhibit translation. And we're going to measure translation rate and things like that. Okay? And in response, the cell dials up ribosome content in a linear fashion. Okay. So we were struck by that. Okay. And then we look at other proteins. Is it just happening to ribosome? Well, no. We look at some catabolic enzymes. So these are the enzymes, the transporters that's responsible for bringing carbon into cells. So in this experiment, we vary the carbon source or titrated the uptake of a carbon, or you go to chemostat and dial the carbon-limited chemostat, dial the dilution rate. Doesn't matter what you do. As you limit the carbon, the catabolic enzyme increases. Okay? And if you fix the carbon source, and you say inhibit, do the same thing, you inhibit the cell with translation by a sublethal dose of antibiotics, it goes down linearly. Okay, and these are very simple experiments that theoretical physicists know how to do. Okay, you have to do grow cell and measure LAC-Z, measure RNA, protein, very simple things. And so you see, this is just a mirror image of that, right? As under carbon limitation, catabolic enzyme increase linearly, ribosome decrease linearly. Under translation limitation, ribosome increase linearly, catabolic enzyme decrease linearly. So then we really have fun then doing, well, what about this, what about that, limiting this and that. Okay, so we did lots of experiments like that. Okay, and always what we get is whatever you're limiting, okay, there's a section that's responsible, that responsible for bringing this nutrient that you're limiting. It increases linearly as you slow down growth. Everything else decreases. Okay, and so all of this behavior can be summarized by a very simple model. And let me describe the model. So imagine this pie, this circle, as all of the proteome in the cell, the abundance of the protein. And among them, a fractions R is used for translation, A is for anabolism, making amino acid, nucleotides, and so forth. Another fraction is the transporters for bringing carbons. All right, so then what this model says is, well, to grow fast, the cell needs to allocate more of the protein for ribosomes, because that's this relation, we know, right? To grow fast, you need to have more ribosome, everything else being constant. To grow fast, you need to also increase more carbon. So if for the same carbon state, to grow fast, you need to have more carbon, linear relation. And I'm not showing this, but you do the same thing for anabolic proteins. There's a similar type of cross. And to grow fast, you need to have more anabolic protein. Okay, so that makes sense, right? You need to have more enzyme to push for flux to grow fast. But then ultimately, this is all of the proteome. Okay, so if you grow too much of this, then you're squeezing out those. Okay. Now, in a simpler scenario, the three of them maybe add up to 100%. But what we find is that about half of the proteome is not related to growth. Okay, so they're basic maintenance that you need to basically express no matter what growth state you're in. Okay, but the basic picture doesn't change. Okay, about half of the proteome, you can shuffle between these different categories, right? And each one is needed for growth. Okay, but they're constrained. Yes?
In all of this, the cell size doesn't change?
So in this case, so the measurements are really simple. That we are measuring the total amount of protein in the bucket. Okay, so cell size is about how you cut the cells. Right? For the question we are addressing here, of course, it's a very interesting question, but for the cell size is not important for this. And also, E. coli being a rod-sized cell, right? The just linear length changing doesn't even, the surface volume ratio is not even affected. Okay. All right. So this simple behavior. So there is a constraint that all of them add up to be around 50%. Okay. The way we know the 50% is that when you look at the finally in the extreme limitation, where it goes to is it doesn't go to 100%, it goes to about 50%. So everything's fixed, right? So just give you, for those of you from engineering physics background, just something to hang your hat on. This system I describe is really very simple. It's just like 3 resistors in a series. Okay, that is in this analogy. So in this resistor problem, the total current going through the system is just in here the growth rate, and the voltage across the resistor is the fraction of the proteome allocated to each function. Okay, so then each one of these relation is this relation between current and the voltage. It's like Ohm's law. Okay, you have several components which is linear. The more enzyme you have, the more flux is going to push. Okay, but the total voltage is constrained to about 50%. All right, so in the system, there's very few parameters. Okay, so these parameters nu C and nu A, they characterize how the quality of a carbon. So like glucose, it's more efficiently taken up, so nu C is larger. Okay, but in these experiments, you don't really see it because this is the variable that's changing. So you don't see that variable. Okay, And so ultimately, there's only one variable in this whole system, that is this 50%, how much is allocated to growth. Okay, so the entire system has one variable, one parameter. Right? So we are getting very hopeful. We started with a problem with billion parameters, and now looking at this in a simple way, we have only one parameter, and we already even know what it is. Okay, so then we try to make some parameter-free prediction. One prediction we can make is for this system is, well, if we change the voltage, right? So if we change this Q fraction, then how would the growth be affected? One way to so this is complicated, but one way to change this is that we force the cell to express some useless protein. Okay, that's not used for any particular metabolic reason or whatever. And then the prediction of this model is very simple. So this axis is how much we express in this protein, right? And so this is for wild type with this none of this. Then okay, I start with some growth rate, right? And then the prediction is that it will just drop linearly until everything is squeezed out to this about 50%. Okay, zero parameter prediction. And well, this is the data. Okay, so our useless protein is just LAC-Z. Okay, we express LAC-Z, and so much that in these cases, 30% of the cell is LAC-Z. Okay, but it still follows reasonably well these predictions. No, it's not perfect, right? But there's no attempt to fit anything. Okay, it's a prediction. But this is true not only for LAC-Z. Then we look back into literature. In the 90s, people were interested in evaluating recombinant technology and expressing all kinds of proteins. Right? And so in some cases, they reported the absolute amounts. So we can make this comparison, and this is the result. So this is for this blue line here. That's for glucose minimal medium, and that's the unperturbed state. And people express, in the various papers, express various proteins, right? And you can collapse them together, right? So notice that these are absolute measurements, right? There's no fitting of anything. Right? This is absolute fraction protein that you measure, right? And this is the growth rate. There's a congruence of this data. So these kind of results make us hopeful that maybe when you look at this, there's some simple aspects of bacterial growth and coupling to gene expression that one can actually make statement that's independent of detail and that can be actually predictive. Okay. So that was 2010, and since then we did quite a bit more work along this line to look at a number of sort of classical problems in bacteriology. And so the number of publications, but I'm going to maybe spend 5-10 minutes talking about one of these problems that was published 3 years ago on the bacterial version of the Warburg effect. Okay, and then I will switch gear to talk about where this simplicity comes from. So let's talk about one application. So Warburg effect is discovered by Otto Warburg in the context of cancer cell proliferation almost 100 years ago. So the point is that energy is most efficiently generated by sending carbon into TCA and burning it off as a CO2, right? But then this is what cells supposed to do in aerobic in the presence of oxygen. But for cancer cells, and also we know that later on for fast-growing stem cells, for example, cells often do not like to do that, but then instead they just do glycolysis and excrete carbon as lactate. They don't bother to use the mitochondria to generate energy. Okay, so why is that? This is not limited to mammalian cells. So Cerevisiae is known to excrete ethanol. This is the beer you drink. And our E. coli excretes acetate, okay, all in aerobic condition. So E. coli is the phenomenon we are studying called acetate overflow. So once again, if you feed it with glucose, and the glucose is burned up completely into CO2, you can generate many ATPs, but instead, E. coli kind of ejects halfway, okay? And it generates about half as much ATP. So this is not the anaerobic glycolysis, so the NADH that's generated can still be traded for quite a few ATPs. Okay, but still, it wastes carbon. It doesn't send it burning off as CO2. Okay. So the question is why does it bother to use the less efficient fermentation pathways? So for a problem that is almost 100 years old, okay, you can imagine that my sort of summary of the state of the fair is any combination of words that can be put together has been put together by somebody, right? An impossible sort of explanation has been proposed, okay? And you think oxygen is available, but actually oxygen is limited, okay? These ecological reasoning. Well, maybe the cells want to just gobble up the glucose and waste the excreted acetate for ecological reason. I mean, it can always be true. You cannot disprove these things, right? And there's a bigger problem behind it. Is basically for any problem that's complex, okay, you can make lots of this kind of a statements, right? But then, so the question is about how do you establish any of these statements? Now, in a traditional field, like so, I came from gene study of gene regulation. There's like a commonly agreed upon rule for the game, right? So to make a statement that this gene is turned on by so and so binding to here and activating something, there are a number of evidence you need to produce, right? Each of which may not be good enough by itself, but together you have array of experiments. Well, then maybe okay, it's believable that something like that is happening. But for a complex problem like that, how do you establish anything? And if we do not have a common agreed upon rules of establishing something, then what are we doing, right? I mean, we can always say whatever. I mean, draw some arrows. There's a little bit of feedback here, a little bit of whatever there. And how do you prove or disprove anything, right? So this problem certainly occurs here. Now, in our study, we came across again a very simple linear relation. Okay, so we look at the E. coli and do our usual thing, feed it with different type of medium, record the growth rate, and you measure the acetate excretion. You find there's a very simple threshold linear behavior. Okay, so I'm going to claim that okay, we have some understanding of where this comes from. Okay, and the way we're going to prove, okay, at least try to validate our understanding, is that since the output is a linear function, right? I know something about it if I know how to change it. Right? If I can do experiment, I can predict if I do this experiment, I can change the location of the threshold, I can change the slope on this and that. Well, then maybe I know something about the process underlying the excretion. Okay, so that's kind of the the approach I'll go by. All right, so let me present. So since there's a linear relation with the growth rate, then we immediately start thinking about maybe proteome is involved. Okay, so here's so first of all the fact, right? So respiration certainly is carbon efficient. That is, you look at these two processes, respiration and fermentation, to generate the same amount of energy flux. Okay, you need a less carbon flux for respiration. Okay, so that's well established, and so you can determine the carbon efficiency. That is, how much ATP flux you get per carbon flux. Okay, and the respiration is more efficient than fermentation. Now, we propose that there's another efficiency, which is a proteome efficiency. That is, how much proteome does it take to push this amount of the flux? Okay, so our thinking was that perhaps the proteome efficiency is the reverse. That is, to generate the same amount of energy flux, we need to spend more protein making the respiratory components. Right? So here's a kind of a cartoon picture to summarize this. Right? So there are two kind of a costs. There's a carbon cost and there's another cost. Let's say, so in this case, the cost of making this power plant. Right? So in a situation where kind of your carbon carbon source is limited, well, then you want to make as efficiently use carbon as possible. and you want to use a nuclear power plant. But if you don't care, you have plenty of carbon, you want to grow fast. You're in a third world country, you want to develop and grow fast, right? And well, who wants to spend all the money making a nuclear power plant? Now you use a cheaper machine. Okay, it's not as efficient, but it gives you the energy you need to grow. Okay, so that's the the balance we're thinking of. Okay, so but then these are just words, right? But the key here is that one is a proposal that is the protein efficiency more cheaper for fermentation. Okay, so that we can measure it. Right? So what we do is we use metabolomics to measure the fluxes, metabolic fluxes. We use proteomics to measure the proteome. Right? And so a lot of work behind it, but here's the result. That is so basically the vertical axis is the measure of the energy flux, and the x-axis is the proteome fraction. And you see, and this is for fermentation, and this is for respiration. Okay, so for the same, if you cut across, for the same energy flux, it takes twice as much proteome to generate the energy. Okay, so that is so that says okay. So this is a precondition for our hypothesis. So it seems like this precondition is satisfied. This is indeed the case for E. coli. But just because it's satisfied doesn't mean E. coli is doing it for this reason, right? So then we okay. So the fermentation is more proteome efficient. It's a linear kind of machine to generate energy. But if the acetate overflow in E. coli due to proteome efficiency, so for that, then we developed a simple model. So it is basically we have carbon flux coming in, and some of it is being used to respiration, some of it used for fermentation, and a lot is used for biomass, and similarly some fraction for protein allocation. And then, so basically, you have a few more equations than the previous model I had. So there are only 3-4 equations. Here we'll have 5-6 linear equations, but very simple to solve. And when you solve them, you get basically a threshold linear behavior. Okay. Now that itself doesn't mean much. Okay, this model produced that result, right? This behavior doesn't mean this behavior is coming from this process. But the nice thing about the model is then now we have a precise prediction, a prediction on how like these threshold points where acetate becomes excreted depends on physiological parameters, and those we can vary in the test. Right? So, for example, if proteome cost is important, then if I force the cell to express useless protein, right, then proteome cost becomes even more important, right? And then, so then we expect cell to excrete even more acetate. Okay, so then this can be done by just doing the same Laxey overexpression construct, and we see that indeed, as we force the cell to make more and more of this useless protein, then this is still aligned, but it's got shifted more and more to slower growth. Okay, and also we can do the opposite. We can get rid of some useless protein that cells naturally express, and see whether this goes the other way. Okay. And so what are useless proteins? Well, in well-stirred medium, motility proteins are useless, right? So we, this is okay. I'm missing a few slides of result. But anyway, so we did this kind of a test. So we knock out the motility proteins, which is 5-10% of the proteome, and you see this shift to the other way. Okay, and then we inhibit it by cells by antibiotics. We see that again ribosome becomes more costly and they expect more excretion. And here's an example where we intentionally induce a proton leak, okay, by expressing some leaky transporter. I can also do this by inducing a decoupling agent, right, to basically make TCA, make oxphos less efficient. Now, normally you might think, well, if I have cell has problem generating energy, then it should turn on more TCA to generate more energy. But in this logic, right, it's anyway comparing what's more efficient to do. And if TCA is less efficient, then it would even use more of the glycolysis to generate energy. And indeed, that's what we see. So if we do either decoupling or we do a leaky transporter, then more excretion is observed. Okay. So through this kind of a experiment, and I skipped the quantity component, right? So the model will predict exactly how much this is shifted, okay? And then the experiment, then you can check how well the model does, right? So through this kind of a study, then we established this for E. coli during exponential growth, right? That it is the interplay between carbon efficiency and the protein efficiency they're doing, right? So the bottom line is that when cells can grow fast, when you're feeding already with a good carbon source, if you want to grow fast, you need to make more ribosomes. That's the growth law, right? You have to make more. If every ribosome is engaged to grow fast, you have to have more ribosome. Okay, there's no other way. But ribosome are making something, right? If you want more ribosome to make ribosome, you have to take their load off something. Okay, so in this case, the taking off making respiratory proteins to make a cheaper protein so that it can make more ribosome and grow faster, right? That's the prediction, that's the logic. All right, then of course, people ask about, well, how does that apply to cancers and stuff? We can discuss these issues maybe informally. Okay, I'll skip that. So let me go to my last topic. How much time I have?
You have time.
Okay.
Let's time for questions. You can take 10 more minutes.
Okay, good. So I want to, so we have done this kind of a application, so to applying the look at classical phenomena of catabolism repression, overflow, this and that, and see, try to understand it in a quantitative predictive ways. Well, and we've been quite successful in applying these very simple linear growth laws to these situations. But then, so the question here we're addressing is, where did it come from? Why? How does the cell know? How does the cell know how fast it's growing and adjust its ribosome just to match what it's supposed to do? Not easy to make cell to draw a straight line, right? If it's some hill function, I don't care. I mean, everything's a hill functions, but straight lines is not trivial. Okay, so where does simplicity come from? So let's look at metabolism in a very kind of a coarse grain bird's eye view. You have a nutrient coming in, and they get turned into a set of central metabolites, amino acid, nucleotides, ATP, and so forth. And then ribosome stitch them together to make proteins, various proteins. Put down some symbols, quantitative. So there's a nutrient flux that's proportional to the amount of a catabolic enzyme that's bringing them. And we have protein synthesis, which is proportional to the amount of concentration of ribosomes. Okay, and then so there's a coefficient here. Then for nutrient, this is the uptake rate of the catabolic enzyme. So in this case, for the ribosome, this is the elongation rate. Okay, so average elongation. Now, pay attention to this. This is going to play a crucial role in what I'm going to say. All right, so of the protein that the ribosome makes, well, some of them, and the ribosome makes all proteins, including themselves, right? So a fraction chi r of the output of ribosome is the ribosome proteins themselves. And then another fraction chi c is the catabolic protein, and similarly anabolic protein and so forth. Okay? So if you look at this, you see this circle here is a self-catalytic circle, right? Ribosome, a fraction of ribosome making a ribosome. Okay? And so ribosome synthesis is really the key to the self-catalytic process, which is basically reflected by the growth rate. Okay, what fraction of ribosome make ribosome? So how is ribosome synthesis, biogenesis controlled? So in bacteria, it is known this is done through a signaling pathway called a PPGP, involving this strange molecule PPGPP, guanine tetraphosphate. So roughly speaking, at a qualitative level, PPGPP is inhibit. So if there's an increase in amino acid content, the nutrient is good, okay, then this pile builds up, and then PPGPP level gets depressed, and PPGPP inhibits ribosome synthesis, and then ribosome synthesis increases. Okay, so the idea is that it looks at its substrate. So ribosome is eating amino acid. Okay, it's consuming amino acid, and it looks at the substrate. If I have lots of substrate, then I make more ribosome to consume the more substrate. Roughly, that's the idea. more closely, if you look at how the details of PBGPP, well, it is synthesized by a protein called the RAL-A, and this RAL-A senses sort of the degrees of uncharged tRNA. Okay. Now we got a problem here. The problem is that there are 60 some uncharged tRNA species. Okay, together with the details of the translation is complicated. Okay, and then it's not clear. So in principle, this control of ribosome synthesis depends on many species of these molecules. Here. Okay, so in other words, if you're the RAL-A and you know any one of the 60 charged tRNA, if they're uncharged, you can slow down growth, right? Which one should you listen to, right? And with what weight? Okay, so it becomes a problem if you try to write down equations to describe this. I mean, it easily involve a couple hundred parameters, just no way to capture this. Now, we might say, well, the cell has a billion years of evolution to fix all the parameters to get this just to the right. But what if I change temperature? Then all the parameters are different. Whether the internal pH level change, all the parameters are different, right? So this is difficult, right? And I maintain that it is not only difficult for us quantitative biologists who are trying to study the problem. It is also difficult for the cell, right? To manage to know what the heck, what it's looking at, right? So the fundamental question we're asking here is, how does the cell know how fast it's growing? Right? What do you look at to know how fast you're growing? It's because all the responses, simplicity, come from it needs to measure. It needs to know how fast it's growing, and then do our response appropriately. All right. So towards that end, so there was so we got inspired by a recent piece of molecular knowledge. I'll show protein structure. So this is a cryo-EM image of a ribosome, the 30 and the 50S unit, and the 3 tRNAs. Okay. Now this tRNA, this is the A position, and this last tRNA was affixed to be uncharged. Right? So there's no amino acid feeding it, so it's an uncharged tRNA. Okay. So it's kind of sitting in a crooked position. Okay. What is behind this uncharged tRNA is RAL-A. Okay. So RAL-A stuck itself into the ribosome. Okay, and then it can only get into the ribosome with uncharged tRNA, because if it's charged, then there will be an EFTU here, and the shape is very different. There's no room for it to go in. Okay. What's more, RAL-A is a big protein. This end is the business end. So this is the end where it synthesizes PBGPP. Okay. So normally it is in a state that's sequestered. So the PBGPP synthesis activity is sequestered. Okay, but when it's in that situation, when you have an uncharged tRNA, right, when the ribosome is loaded with the uncharged tRNA, right, then it does present an opportunity for it to give a pulse of a PBGPP. Okay, so looking at this structure, so this inspires us to think that maybe what this is telling us is that PBGPP, or the RAL-A activity, it's sensing the degree of activity of the ribosome. Okay, so basically ribosome, if you have a charged tRNA going in, then it's possessive, and it goes to the next step. Okay, but then it stalls the waves when you have an uncharged tRNA going in. Okay, so then it's a way to sense the activity or the elongation rate of the translation. Okay, so that's kind of the inspiration we got. That is the big pools, hundreds of molecular species that goes into determine the elongation rate. It's not directly sensed, right? But what it's sensing is just through the elongation rate itself. Okay, so instead of instead of having this big pool of molecules controlling the ribosome synthesis, right? This structure to us suggests that this may be done by a process where it's the elongation rate that affects the synthesis of PBGPP and the ribosome control, right? And the elongation rate itself, of course, depends on all of these molecules. Okay? So what is it suggesting? Mathematically, what this means is instead of depending on hundreds of variables, it depends on 100 variables through a single variable, okay, which is just the trick we had mentioned before in statistical mechanics. Okay, so this is a entry point where dimension reduction can happen. Right? So, taking this hint, then we use this relation as an answer. That suppose the ribosome control is done through by the cell measuring its own elongation rate. Right. Then we can very straightforwardly write down a few equations describing the control of the synthesis of ribosome and catabolic enzyme in terms of this elongation rate. Okay, and the elongation rate it can be determined through the ratio of these concentrations, and we end up with a unique ordinary differential equation with no free parameters on the state of elongation. Okay, so also there are two regulation functions which can be fixed by the steady state properties, because in steady state with the growth rate we know everything. Okay, so there's some regulation function we can deduce. So with this equation, one can go ahead and solve for the time dependence of the ribosome, catabolic enzyme, growth rate, and everything. And so that's the result I showed you at the beginning. Okay, you just feed in you have to tell the system initial condition, because you have to tell where you're starting from and where you're heading to. Okay, in terms of the nutrient, and it sort of figures out everything else. It's a completely constrained system. All right, for upshift, for downshift, and we also looked into the proteome itself. With the proteomics, then we measure what proteins go up, what proteins go down, coarse-grain them into several sectors. So the lines are predictions, and the symbols are data. Similarly, for downshifts. All right, so that's the application of this scheme of dimension reduction, but then the key here is this relation that the change in translation leads to a signal that the ribosome synthesis is regulated by sensing the translation rate. Okay, so this is still a hypothesis, right? What I showed us the application, taking that as a hypothesis, it leads us to good things. Okay, now we're directly trying to verify this by doing lots of difficult sort of challenging experiments to see whether they're really related, in real time. Okay, so that aside, so I want to make several comments on this particular mode of a regulation. Right? What this regulation is, it's a regulation based on activity. Okay? Now, in biology, we are most familiar with regulation based on concentration, right? So the most frequently talked about is the end product feedback inhibition, amino acid synthesis, end product go back inhibit. Okay? So this, and the other type is a kind of a supply-driven. If you have lots of a substrate, lactose, a lot of lactose, and the lac system is turned on and taking lactose. Okay? These are the two common ways of regulation we're used to. And these schemes of regulation work very well when you have a well-defined sort of input, well-defined output. Okay? But here we're dealing with a problem, right, with many, many input, many, many output, right? And if I'm going to say that the regulation here, supply-driven or product inhibited, that regulation scheme based on concentrations is a curse when you think about complex regulation. Why? Because every time you talk about concentration, you have to talk about all the parameters that concentration brings in. Right? So you have to talk about the KMs when you talk about concentrations. Okay? But in a complex dynamic process, what's the significance of a particular value, of KM, right? In fact, I would go even a step further to say that the way we are taught, certainly the way I learned molecular biology, biochemistry, right? Everything is based on concentration. Biochemistry is based on concentration. Okay, so it's very natural to think of a regulation or in terms of concentrations. But, right, because of this parameter explosion problem, as soon as you talk about concentration, you have all this problem, the complexity, and I would like to think of the way to concentration as a trap. Okay, in the sense that when you think about gas, studying property of gas, right? If you're coming from a Newtonian way, thinking about individual particle, it's very natural to think about trajectory of a particle, the position and the momentum of particle, to even think about gas. But as soon as you think about that, you're lost. You never get the property of the whole. Right? So I'm suggesting that maybe there's something more, that nature maybe not as comfortable as we're thinking about a concentration-based regulation. Right? Maybe for complex systems, another clever way to think about it is really look at the flux control, activity control. Because then you get rid of the parameters. Okay? So to sum this up, so imagine you're running a big factory, right? So you have having a lot of workers making lots of products, right? You want to decide how many workers to hire. What do? So in the supply-driven or product inhibitor way, you can have an inventory list. Oh, what sort of inventory is very low? What products are being built up? And you have a table of who is working on what, right? And then try to decide which person to hire, which person to fire. There's an alternative way, okay, which is much simpler, which seems to be what E. coli is doing, at least for the growth control. And this way is simply to look at the number of idle workers. Right? If you see a lot of workers taking break, you can fire them. Okay? Right? And if nobody's sleeping, well, you better hire some more workers. Okay? So that's right. So you don't need to know the detailed parameters, and just look at the activity of individual workers. Okay, use it or lose it. Right? So to put it into the context, right? This is one small example that we have spent quite some time working on, bringing molecular sort of interaction and the physiology together to try to put it into a predictive framework. Of course, the eventual goal is to look at a more complicated system. Right? Well, so we'll have certainly plenty of work to do together. So let me acknowledge the people that did the work. The overflow metabolism was done by a postdoc, Marcus Basson, who is now an assistant professor at the Harvard Medical School, and Tony Hui, who's just finishing up his postdoc in Princeton, so is on the job market now. And then the work on growth transition kinetics that's more recently by two graduate students, David Erickson and Severin Shrink. Okay, so and overall, our goal is for quantitative predictive understanding of the organizing principle of living systems. Thank you.